Every year, millions of people are trapped in the diabetes dilemma. Type 2 diabetes and obesity, two diseases feeding each other in a cycle of pain and dependence. Current treatment, from insulin injections to dual-target drugs, have changed lives but remain invasive, temporary, and burdened with side effects. The question remains, can we do better? As iGEM, we ask, what if light itself could heal? What if we could control insulin and GLP-1 precisely, reversibly, and without pain? The glowing blue light expands into our project LEGO. LEGO, light-induced expression of GLP-1 and insulin. Now introducing LEGO, an optogenetic gene therapy platform that uses light to control dual hormone secretion, smart, non-invasive, and personalized. A new way to break the diabetes loop.
This brings us to the design of our solution, the LEGO system. The name LEGO captures its essence, light-induced expression of GLP-1 and insulin for obesity and diabetes treatment. Our vision is a therapy that is optically controlled, orally administered, and dually beneficial. The system operates through a sequence of 5 key steps. First, the patient orally takes FFC, which enters target cells and serves as the substrate for nanolug. This enzymatic reaction generates intrinsic blue light, turning each cell into its own light resource. This intracellular light then activates the optogenetic switch and pairs up iLEAD and SSPB protein. Their precise, light-dependent binding initiates downstream signaling. The iLEAD SSPB complex then recruits the transcriptional activator GIL4-VP64 to promote her 5-tandem US repeats, triggering the expression of our therapeutic genes. The light-induced iLEAD SSPB complex then brings the transcriptional activation domain VP64 to the synthetic promoter, which is pre-bound by the GIL4 DNA binding domain, thereby initiating the expression of our therapeutic genes. Finally, using an iris component, the translation simultaneously produces 2 distinct proteins from a single transcript: insulin for glycemic control and GLP-1 for appetite regulation. This integrated mechanism confers 3 advantages. The route of administration is non-invasive, relies solely on an oral pill. The trigger is precisely controllable, as therapy is activated only by the internal light signal, and the outcome is dual beneficial, concurrently addressing both type 2 diabetes and obesity through a unified genetic circuit. A robust theoretical blueprint, however, must be validated by experimental data. Our wet lab work thoroughly demonstrates the functionality of the design.
We translated our LEGO system blueprints into reality through 3 interactive design, build, test, learn cycles. The light source module, the optogenetic switch, and the dual hormone expression module. Each module was experienced through the process of learning, design, build, tested, and finally come back to the stage of learn. Cycle 1 is about validating the optogenetic switch. Let's walk through the experimental journey. We started by testing the iLEAD SSPB optogenetic pair. By tagging them with fluorescent proteins, we confirmed they remain stable in the nucleus under prolonged blue light, unlike other systems like Cry2 that leak out. The switch was also rapid and fully reversible. Upon light exposure, it induces dimerization within seconds, and the proteins dissociate immediately once the light is turned off. This provides us with a precise and reliable optical switch. Cycle 2 is about bioluminescence activation. Given the limited tissue penetration of external light, we introduced nanoluciferase to generate blue light inside the cell. By fusing nanoluc with iLEAD and adding FFC as the substrate, we achieved rapid protein recruitment comparable to that induced by external blue light. This breakthrough enables fully non-invasive endogenous activation. Imagine triggering the system simply by taking an oral pill, paving the way for future in vivo therapeutic applications. we went from stuck with axonolite to unlocking internal bioluminescence power. And that's what makes the synthetic biology so exciting, turning flaws into breakthroughs. Cycle 3 is about dual hormone expression and bioactivity. Finally, we fully assembled the LEGO system to enable universal secretion in mammalian cells. We engineered the hormones by incorporating a furin cleavage site into insulin and a degradation resistance AAG mutation into GLP-1. This allowed us to achieve precise, light-controlled co-expression of both bioactive hormones. We demonstrated that blue light significantly enhances both mRNA levels and secretion of these hormones. Most critically, our biosynthetic insulin proved fully bioactive. It effectively activates downstream PI3K-AKT signaling pathway through the insulin receptors, confirming its therapeutic potential. Our wet lab conclusion is about through 3 interactive DBTL cycles, we robustly activated and validated every module, proving the LEGO system is feasible, reversible, non-invasive, and functional. We successfully established 2 activation pathways: the convenient oral bioluminescence and the precise external light illumination. With the systems functionally confirmed, our dry lab teammates will now focus specifically on optimizing the external light pathway to answer, how can we make it work even better?
While our wet lab successfully built the core LEGO biological system, we recognized that translating this innovation into clinical practice required a dedicated interface. This led us to the creation of our dry lab component, with the core objective to bridge our optogenetic therapy with real-world patient care through integrated software and hardware. Dialight represents the evolution of our experimental light control system. Initially developed to provide precise blue light simulation for our wet lab experiment, this hardware platform has now been refined into a compact, wearable device. Built on a low-cost, compact ESP32 microcontroller, it creates its own Wi-Fi hotspot. Through a simple web interface, users can receive and execute the personalized light regimens generated by Diaplan, delivering precise blue light simulation. The development of Dialight promoted a crucial question: how do we translate complex patient data into optimized light prescriptions? This led us to create Diaplan, the intelligent brain behind our light delivery system. Diaplan is our web-based software that acts as a precision dosing engine. It integrates patient data like weight and blood glucose to calculate personalized doses for both insulin and GLP-1 combination therapy. Its core innovation is the dose-to-light translator, which converts the biological dose into precise blue light parameters, intensity, and duration, required to activate the LEGO system. Together, they form a seamless, closed-loop treatment system. The patient uses Diaplan to generate a light prescription, inputting it into the Dialight wearable device, which delivers the exact light pattern. This light then activated the implanted LEGO system, triggering the production of insulin and GLP-1 inside the patient's body. This completes the full circle from digital calculation to biological action, making personalized, non-invasive gene therapy a practical reality. This dry lab component is how we ensure the LEGO system is not just a lab wonder, but a viable, user-centric therapy ready for the future.
This is our human practice journey, a structured, iterative process that fundamentally shaped our project evolution, ensuring every scientific decision remains deeply connected to real-world needs and clinical realities. To build a responsible project, we executed a full-cycle human practice strategy, from global data research and a 427 people patient survey that define our core mission, to expert interviews that pressure test our design, and public education that build essential trust. It was an in-depth conversation with patients like Mr. Song that made the data come to life, revealing the profound human needs behind the statistic. This direct human connection, combined with our survey data with over 80% struggle with the injection and 72% need to combo therapy, combine our validation. It directly shaped our 3 pillars: oral activation, dual hormone output, and spatial temporal control to minimize side effects. We were engineering a response to a deeply human problem. But design must meet clinical reality. An endocrinologist challenged that a fix like those was dangerous. Our response: we engineered the diet plan software and diet light hardware to enable personalized dosing. A translational scientist pushed us on scalable manufacturing, guiding our early R&D strategy. And insight from patient interviews reinforced our focus on a seamless, life-integrated design. Even our molecular biology was refined through iGEM collaboration with HiZJU China, adopting a GlyA substitution to enhance GLP-1 stability based on their shared insights. Beyond the lab, we engage in sustainable education through our blog and social media, demystifying SymBio for thousands. We analyze the competitive landscape, identifying a clear market needs. Synthesizing all these feedback from the patient heartfelt need to be clinician rigorous demand. LEGO evolved into a holistic ecosystem, an implantable device activated by a pill or our personalized software. All design was built in biological containment for safety. Every conversation, every piece of data, every human story left its mark on our project. This is what integrated human practice means to us, letting the world write the specs for a better therapy, one that answers not just biological needs, but human ones.
Cut between real job footage and stylized graphics. One visual for each problem solved. First, injection, replaced by glowing light. Second, dual target animation. GLP-1 plus insulin waves are intertwining. Last but not least, remote control dashboard, data visualization, precise hormone release. Step by step, LEGO answered each challenge. Non-invasive activation through light, eliminating the pain of injections. Precise, reversible control through our optogenetic design. We target both diabetes and obesity simultaneously. But LEGO is just more than biology. It's a system.
Our modular software architecture with adaptive dose calculation and API integration lays the foundation for the software tool. Our integrated human practice turns expert insights into design, ethics, and real-world readiness. Together, these form the composite heart of LEGO: science, software, and society fused into one. And this is only the beginning. Our next steps: animal experiments, device encapsulation, and collaboration with industry, bringing LEGO closer to real-world therapy. But imagine going beyond diabetes. Imagine using light to program cells to produce any therapeutic protein safely and on demand. One platform, infinite possibilities, a brighter programmable future of medicine.
Ladies and gentlemen, welcome to listen our team song. 3, 2, 1. GLP-1, it starts to rise, insulin flows, no compromise. A dance of self, decoded me, with science and life and needs. Shine alive, let go emotion, healing